we had actually just finished up talking about lipids, which, as we noted, were surprisingly strong in variety for function. So what we're going to do is you're going to pull out that handy-dandy in-class activity, okay, and we're going to add a second line for lipids. Under lipids, you're going to have 2 bullet points again. Bullet point number 1 and bullet point number 2. Okay, note this time, because lipids do so many things, that you have to give me a list of 2 things that they do as your first bullet point. Okay, your second bullet point can still just be one way that you would identify a lipid or tell the way a lipid is different from all our other biomolecules. Okay, and remember, as we're thinking about lipids, in this case, we can really focus on how would I know a lipid is different from a carb, because we already finished carbs. Okay, so make sure this is going on the same piece of paper that you did your carbs before, or in the same file. Right, and then make sure if you're doing it in a file that you save it afterwards, because we're going to continue to add to this. Bless you. And remember, it's okay to flip back and look, but do your best to try to answer it without looking first. This is a really good study technique. That way you're kind of forcing yourself to try to recall it, and then you're only using your notes to check. Okay. Any questions about lipids or the differences they have from carbs before we move on to our next and most favorite of all the biomolecules? That's good. Not necessarily that they're quiet, but that there's no confusion. All right. Back on Wednesday. Okay, so as we move forward then, our goals remain the same, okay? We're still going to try to focus on what is the point of this biomolecule and what function is it serving inside the cell or tissue or organism, and how can I identify, gracious, I just need to go back to bed today. How can I identify this biomolecule as it differs from all biomolecules? Okay, and now we can directly compare it to lipids and carbs. Okay, and as we'll find, proteins are our favorite biomolecule, aside from being delicious. Okay, these do the most. Okay? The most variety and function out of all the biomolecules that we're going to talk about. Okay, that's saying something because lipids have a pretty big variety and function. Okay? But proteins have even more. We're going to see these come up a lot over the course of our semester together. So this is just a general list of all the things that proteins can do. All right, so proteins make up the core of enzymes. You'll recognize an enzyme over the course of the semester because they generally have an ACE ending. So lactase is an enzyme. Is an enzyme that breaks down milk sugars. Okay. Proteins work for storage and structure. Okay. We eat protein to help build muscle. Okay. Proteins are a lot for transport. We're going to talk about these a lot. Someone asked a great question last week about how do you actually get things across the plasma membrane. Proteins do that, right? They make channels. They're kind of like tunnels under bridges. And they work hard to make protection and regulation. They make other walls. They regulate how things come in and out. Okay, so proteins will come up a lot as you continue going forward. If you're a biology major, you'll talk a lot about proteins. In anatomy and physiology, they deal a lot with protein ion channels. They make your heart pump and muscles work. Right, so proteins are super important. So what does the protein look like? So we're going to spend a lot of time talking about proteins because proteins are so versatile. They have to be really flexible in the way you make their shape. So this is the smallest subunit of a protein, aka an amino acid, which you've probably heard that word before. So the amino acid subunit has three parts that we care about. Okay. The amino subgroup, what you have on the left-hand side. Okay. The carboxyl subgroup, that looks familiar, right? That one we've seen before. And then the R group, which I have in the blue box, which in the past we've just defined as other stuff. You know, for once, as we'll find, this other stuff is actually going to matter. We're going to stick that on the back burner for right now. And so let's focus on our two red groups right now. All right. So let's look at the right-hand group, our carboxyl group first. I always like to start with what's familiar. And our carboxyl group is super familiar. Okay? So this red group, in fact, both red groups, are always the same on every single amino acid. Okay? This is unchanging. Right? And we've seen our nice carboxyl group before. What other molecule has a carboxyl group? A lipid. Good. Okay, well, that's not great, I guess. So now we found something that's similar with another compound. Okay, so proteins and lipids both have carboxyl groups. What do carbs have? Carbonyl, good. Ooh, which? looks like this, right? Spelled absolutely terribly. That's why I make slides. I don't spell on the fly well. Okay, so that's one difference. Carbs have a carbonyl, okay? Proteins and lipids, okay, have this carboxyl business. All right. Now, because lipids and proteins both have a carboxyl, we need something else to tell them apart, right? That's not enough. So that's why we have this left-hand side to focus on. This is brand new. We've never seen this before, right? So this is the namesake of our amino acid, the amino group. And this looks super different than anything we've ever seen. So this has a nitrogen in it. This is the first compound we've seen that has anything that has a nitrogen in it. And it's only going to be one of two that we're going to talk about that does. And the other one looks way crazy. So it's going to be a little easier to tell it apart. Okay, so our amino group looks pretty simple. It's just a nitrogen, two hydrogens. Okay, it's a very stable looking, okay, well-dressed, business-like compound. Okay, so the structure is not too bad. It's got a carboxyl on one wing, amino on the other, and it always is going to look like this. Now this in and of itself, though, we know isn't enough because I just got them saying that proteins do a bajillion things. So if you're going to do a bajillion things from building walls to tearing down other compounds, you need something that's going to give you a lot of variety. Okay? Structure makes function. We're going to focus on this a lot as we go. So I'm going to drill that into your heads. But if structure equals function, man, this is the same. So we need something that's going to give us that variety. And the something that's going to give us that variety is that R group. There's something else. Okay? So if you've ever talked about amino acids before, okay, there are a whole bunch of them. Okay, naturally speaking, in fact, there's 20. 9 of which you need to survive. This was the core plot of Jurassic Park, in fact. Right? They took out lysine from the dinosaurs, and then they gave it to them specifically in their diet. So the idea was if they escaped from the island, they would all just die. Worked great. But that was the concept, right? Lysine is an amino acid that you have to have in order to survive. So the way all these amino acids are different and perform different functions in the body is they're all slightly different in this other group. Okay, with me so far? All right, so here's how they're different. Don't freak out. Okay, I don't care that you memorize this. Again, this is not biochemistry. And here's what I do care that you understand about this. Okay, we still have our wings on here, right? Even though they're drawn a little funny. Here's my carboxyl group. There should be an H there, but this is chemistry. Right, here's my amino group. Okay, so all the colored stuff on here are my R groups. Okay, so this is all the different stuff of which you will never, in a million years in this class, have to memorize or recognize and name. Okay? So what's important and what I want you to know is that they're different. Okay? And the fact that they're different, we're going to use to get different interactions. And this is what's going to make their different shapes. And we recognize these different interactions already with what we know. Okay, so these side chains are nonpolar. Okay, the green bits that I have highlighted here. Okay, what else do we know that's nonpolar? one did we know that was nonpolar? The lipids, good. Right, those phospholipid tails in particular. What happens when you put two nonpolar things together? What happened with our phospholipid tails? Did they ignore each other? Did they attract each other? Did they reject each other? They attracted each other, right? Okay, remember our two little phospholipid tails came together and kind of created that little closet? Okay? So we already know that happens. So we would expect nonpolar things to attract or hide. Okay, so we know what kind of interaction we're going to get there. Okay, let's float down to the bottom here. Okay, those are electrically charged things. We haven't talked about those in this class, but we can kind of guess. Okay, so we have a negatively charged set here in pink. Okay, and a positively charged set in blue. Okay, so if I have magnets, how do those work? Am I going to get a negative and a positive or two positives together? Opposites, right? So I would expect a negative and a positive to hook up. Okay, so this is what's going to be really important here, is that within all of this business, okay, there's going to be a whole bunch of different interactions within these side chains. So depending on which amino acids are present, whether you have some negatives and positives or some nonpolar ones, okay, whether they're sulfides, a whole bunch of stuff you don't necessarily need to know. What you do need to know is, depending on who's there, you're going to get different interactions, different bonds. And when different things hook up, you'll get different shapes. Okay, that makes sense?
So we had a question in the chat really quick. So they said, will an R group always be referred to as an R group, or can there be a different name?
So they'll be referred to as R groups or side chains, and that's it.
And another question was, do all amino acids have nitrogen?
All amino acids do have a nitrogen. They always have that amino group, yes. That's always their left wing. All right, and we can see that here. They're drawn kind of funny. Because these are all drawn here to be bonded, which is all stuff you guys don't care about right now. But we can see that they're all there. Okay, really good questions. Any other questions about what we would expect an amino acid to look like and the wide variety that we have and why that variety is there? And we'll focus on it some more because we're actually going to do some things with these side chains. Cody. One more time for me. You mean the side chain? Yeah. So I'm never really going to ask you that. In this case, I've just told you that there's a pool of negative and positive ones. I'm never going to make you identify a side chain. All right, so in this case, the way you would know is we're just focused on the stuff that's in the red and blue. So our negative ones are here, and the positive ones are here. And so that's where that's coming from. But it's not something I would make you dig out and identify because that's not what we're interested here. And you can see that those are lacking in the side chains here. Okay, any other questions about what makes an amino acid, which is the smallest component of a protein? Okay, so we have the amino ring, the carboxyl ring, and then the wide variety of R groups. Well, I can't spell anything today, guys. I feel okay? Let's look at that closer then. So, like before, like we did with our lipids, okay, like we did with our carbs, okay, when you want to make bigger chains, we can combine them. Okay, so okay, if I have two small amino acids and I want to make a large chain or one large group, am I using hydrolysis or dehydration? Oh, I have it on there. Rats, that's no fun. Okay, so remember we're using dehydration synthesis. Okay, so we have 2 small amino acids, and we're going to make one large connected group, which is actually called a dipeptide. We'll talk about why that is. Okay, just like with lipids, all right, we like to use new names, but we're still following the same process. Okay, let's focus on the very top here. Okay, not let this slide get us. So let's look at amino acid 1, aka Jane. Okay, Jane has some very familiar equipment. Okay, she's got our carboxyl group. This is the exact same equipment that we used when we made our lipid, right? Okay, and our carboxyl group has the same stuff we've had to use before. Okay, our hydroxyl. And we've always used a hydroxyl to do dehydration synthesis because remember our goal is to make water. Okay, so thank you, Jane. So we're going to make Jane a little stumpy. So there goes our oxygen and our hydrogen. Okay, so this is one place this is going to look a little bit different. So Carl does not have a hydroxyl on his other end though, right? Normally we would have a hydroxyl sticking off the other side. We don't have that this time because we have a brand new left wing, that amino group. Now that being said, why this is different, okay, with that shiny little nitrogen sitting there, we still have everything we need, right? So we have our oxygen and we have our hydrogen already. That's 1 oxygen and 1 hydrogen. To get our water out of there, the only thing we need left is 1 hydrogen. Our amino group has hydrogens. Okay? So there's no reason we can't use our amino group to hook up. That works. Okay? So we're going to take that hydrogen off our amino group. And there's our water. Okay. So the only thing that's different here is now we've made a very unusual bond. Okay? Because now we've made a connection. Our handhold isn't between a hydroxyl here. It's between the carbon and the nitrogen, which is kind of weird. Okay? But we haven't done anything we haven't done before, right? We still took out an oxygen, hydrogen, and another hydrogen. We've just done it in kind of a weird place. Okay, so there's our carbon-nitrogen bond. So we lost the hydrogen here, right? And we lost our hydroxyl group there. So because we've changed from making a connection between a carbon and an oxygen to a carbon and a nitrogen, okay, we put a new hat on, new hat, new name. Okay, so our new name is a peptide bond. Okay, calling something a peptide bond indicates that you have nitrogen in there as the core of the, gracious guys, I'm so sorry, as the core of that bond. Right? Remember, we were a covalent bond, we were C to C, oops. We're an ester bond. We were that C to O to C to double bond to O, right? We had our little hat there. Ester bonds from our lipids. So our process is the same. We're still using dehydration synthesis. We've still used a hydroxyl off of our carboxyl. All of that's the same. Okay, we've still stolen a hydrogen. We just stole it from a weird place. We were creative with our process. Okay, and so doing stuff like this is the reason why it was really important to understand the process of dehydration synthesis.
Can a peptide bond link through hydrolysis too, or is it always dehydration synthesis?
So you'll break it by hydrolysis, right? So yeah, we could absolutely clean this up a little and go the other way. I don't have a slide to do that, but we can do it. So if we wanted to go the other way, you're absolutely correct. So we can break a peptide bond if we wanted to break Jane and Carl up. Okay, so our goal is to snatch-tastic that, right? Okay, we just have to be specific in this case about where our oxygen is going. Okay, where in the past we could be kind of loosey-goosey, okay? Because for Jane, okay, which is the side that has the carbon, okay, we have to convert that back to a carboxyl group. Okay, so Jane in this case 100% has to get the lion's share of that water. Okay. So Jane is going to get the OH from the water. Let's go ahead and change colors here. And Carl will get the leftover H. And that hydrolysis will break that peptide bond and give us two separate amino acids. Okay, great question, okay, and perfect application.
So there was another question, and they asked, does the type of bond affect the strength of the bond? So it's like peptide strong.
So generally not really. They're all ultimately sisters of the covalent bond. At least not in any very dramatic way. Not like ionic bonds are stronger than. You're not going to see an hierarchy like that. That's a great question. Okay. So this looks really unfamiliar. It feels a little intimidating when you're first looking at it, and so really take the time to stop and work through the process and feel where the pieces are the same. We're still taking that hydroxyl group. We're still taking a hydrogen and making that water. Okay? The bond is still there. We're just using a new element to do it. We're going to call it something else or put a different hat on. But ultimately, we're following that same process that we did on slide 3 or 4, okay, way back at the beginning. Okay, really great questions. Any other questions on this? So the key here is as we go to make our proteins, right, the amino acid is sort of the core subunit. And we're going to take and make polypeptides, right, a long chain of these amino acids in order to make a protein. Okay. So in order to make a protein, we need to link things together at what we call many levels of organization. So everything we've talked about up to this point is pretty much just the primary level of organization, linking together those amino acids. In order to make a functional protein, we're actually going to need to twist and fold this multiple times to make a three-dimensional structure. And remember, the ultimate three-dimensional structure for this is really going to depend on those R groups. Okay. So what kind of bonds we can make and where they are. And we'll actually get to see that and sort of explore that. All right, so let's look at these levels of organization. Please. Thank you. All right, so the primary level is what we just spent all that time talking about. Okay, I like to call this like the chain of pearls. Okay, so this is just the amino acid sequence. Okay, we take amino acid 1, amino acid 2, dehydration synthesis. And we're going to take a third amino acid, dehydration synthesis. On and on and on and on. We can get a lot of variety doing this, right? So remember, we have 20 different types of amino acids that are all interesting and unique that we can lock together into a chain. Okay, so each of these round dots on the screen here would be a different amino acid. These all have different R groups potentially attached. Now, obviously, there's going to be some repetition, right? Because many of these are going to be hundreds or thousands of links long. Excuse me. Right, but you can ultimately get a lot of variety. And remember, this is held together by those peptide bonds. Okay, the ones we created via dehydration synthesis between that carbon and that nitrogen. But the key feature here is this is very flat. Okay, it's like taking a necklace and laying it out on the counter in front of you. I have a camera now. This is going to take a lot to get used to. Get me out of the PowerPoint. Thank you. So, from here on out, Jasmine, I might have you flip back and forth. All right. So, because that went terribly. So we want to think about having our string of pearls, so it's going to be very flat. Okay, so very two-dimensional. We have a length and then the width of our amino, from the carboxyl wing to the amino wing. Okay, but there's no depth to it because we haven't twisted or folded anything yet. So as we go, right, it's going to take us several stages before we make kinks and twists to get this folded up to a three-dimensional structure. Okay, so that's familiar. That feels okay. Feel good so far? Okay, let's look at the secondary structure then. I'm going to draw in here really quick. Sorry about that. Okay, so the secondary structure is going to start to put kinks in your flat necklace. Okay, so our kinks are going to occur as hydrogen bonds. Okay, so I'm going to never think that these are very big. So if we think about what's left over from what was our carboxyl group and was our amino group. Okay, this is what's key here. So what's left over from our carboxyl group, right, we took the OH, the hydroxyl part away. Okay, that was taken away during dehydration synthesis, but we still have the double bonded O, right? We all remember that from a couple slides ago. That's still there. And from our amino group, we took the nitrogen. Okay, we took one hydrogen away, but we had two to begin with, right? So we have one hydrogen left. So hydrogen bonding, okay, has to do with the selfish roommate rules. So remember that our oxygens, when we talked about this way back, are just a little bit negative, right? Because they're selfish and they steal electrons, kind of like your selfish roommate uses too much toilet paper. Remember talking about this? Right. That's still the case here. And so that makes our oxygens and our hydrogens attracted to each other. And there's a whole bunch of these floating around, right? So there's a bunch of double bonded oxygens left over, abandoned, the remains of our dehydration synthesis. And there's a whole bunch of single hydrogens left over. Okay, it's like singles night. So all these guys are remaining. Okay, so you can see the double bonded oxygen, the single hydrogens hooking up. Okay, throughout our structures. So anytime they get close enough together, okay, they're going to hook up. And these bonds are pretty strong. Okay, so depending on where they are when they hook up, will you flip me to the other camera?
I don't think it's gonna let me.
It should let you.
It should let me.
Mm-hmm.
So you're gonna go to the share matrix.
Switch to share camera. Switch over to view share camera.
Yeah.
Man, that's such a good idea. That's okay. So what ends up happening is we have our flat plane, and those features start to twist. Right? So we have our alpha helix, which are rolls, and our beta pleated sheets. I didn't name these things. Okay, alpha just means one, beta just means two. Okay. Okay, beta pleated sheets are just kinks. Okay, these are still flat though. Okay, so all we've done is taken what was 100% flat and twisted it, but we still don't have a good depth to it. Okay, we basically just rolled our flat thing over. Okay, we've created a nonlinear shape, and that's really important. Okay, but we don't have any depth, right? We're not going up in any particular direction. Nope, can't just click that. Share back to this. That work? Okay. 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 Now, any given structure is going to have both the helix, right, the twists, and the sheets, the kinks. Okay. Because remember, this given polypeptide is thousands of amino acids long, right? So you're not just going to have one long spiral. You'll have maybe 100 units of a spiral, then 100 units of kink, then another 100 units of spiral. Okay, so it kind of looks like an old school hair dryer that can't seem to get it together. Okay, any questions about our secondary structure? Okay, we're going to go ahead and stop there, particularly because it's taking me so long to flip back and forth between the camera. Not for lack of trying, mind you. Don't forget that Jasmine has the socrative up. There is one last thing I want to remind you of while you're filling out your socrative. Fallon's study session is tomorrow at 6 p.m. And it'll be in that same, or if you haven't been there yet, there's a special study session, collaborative. Okay, so make sure that you fill out the socrative, last name, first name for your name. The teacher's question, of course, is the slide we finished on. Then once you've finished up, the back two sections can go out the back door. Thanks for sticking it out with me, guys. I know dealing with this tech can be a little frustrating, and I always appreciate your patience.
So one of the kids pointed out in the chat, like this is what it looks like for the kids, and you're sharing your screen. I think you can just click that and see that.
I actually didn't know they could see me.
Well, it's very, very small, but if you just tell them to click that, you don't have to flip back and forth. They can.
Wow.
I have no idea that was possible, but Haley, she pointed that out in the
Because when
chat.
Dr. Turner and I demoed this, this might be updated because there was no response.
Yeah.
So I wasn't aware they could see me while I was running the screen share.
Yeah, I knew that they could fill out the little box, but I didn't know that they could.
That's really cool.
So, for future reference, you don't have to.
Yeah, that's actually way better.